Hi, today I'm going to talk about cloning. But first of all, what is cloning? It is the process of producing similar populations of genetically identical individuals that occurs in nature when organisms such as bacteria, insects, or plants reproduce asexually. Dolly the sheep comes to mind when we think of cloned animals. She was made famous as the cloned sheep made in 1990, but the first animal ever cloned was in the 1800s. As for humans, the first hybrid human clone was created in 1998. By advanced cell technology, it was created using SCNT, somatic cell nuclear transfer. A nucleus was taken from a man's leg and inserted into a cow's egg from which the nucleus had been removed, and the hybrid cell was developed into an embryo. There are three main different types of cloning. The first one is called DNA cloning, when only the DNA of a cell is replicated. A DNA from an organism is transferred to a self-replicating genetic element such as a bacterial plasmid. In other words, a small piece of the DNA component is removed and united with a plasmid which reproduces itself to create multiple copies of the same DNA code. This copied DNA can then be spreaded in a foreign host cell. After it is introduced into a suitable host cell, the recombinant vector can then be reproduced along with the host cell DNA. The second type is reproductive cloning. It is a technology used to generate an animal that has the same nuclear DNA as another animal currently or previously existing. Human cloning also falls into this category. Dolly was created by reproductive cloning technology in a process called SCNT. The DNA information from the nucleus of a donor adult cell is copied into a cell whose nucleus has been removed. Chemicals or electric current are used to stimulate cell division. Once the cell starts dividing and the embryo reaches a suitable stage, it is planted into the uterus of a female host where it develops until birth. The third and the last type of cloning is the therapeutic cloning. It is like reproductive cloning, except that the embryos are not allowed to develop fully. Its purpose is to extract the stem cells from the embryos and study them. When the egg had been cloned and divided for 5 days, the stem cells are extracted from it, and the embryos are destroyed due to the extraction process, which raises ethical concerns. One more thing, even though the DNA from the nucleus in the cloned cells is identical to the original one, the whole animal or human is not identical. In other words, the result is not a twin. The answer of this question varies from one country to another regarding their culture, tradition, and religious views. For example, cloning is illegal in Canada, Australia, India, and Colombia. However, it is legalized in other countries like Greece, Portugal, and Ireland. People against cloning have numerous objections about it. Firstly, it would contradict the most fundamental principle of medical ethics, that no human life should be exploited for the benefit of another. They think that human cloning must be banned now to prevent state-mandated eugenics in the future. From a more liberal standpoint, they worried about equality if inequality is designed into people's genes. If an ordinary child and a genetically enhanced one are competing for the same liver transplant, who gets it? Second objection is the therapeutic cloning, for it requires a large number of eggs. On one estimate, you might need eggs from 80 million women just to treat one diabetic case. This fear resonates not just with the religious right, but also with the feminist left, which worries that women will be sucked into the embryo business unwillingly. For the third objection, they called it the slippery slope. Legal therapeutic cloning, or some critics, would produce stockpiles of cloned embryos for research. Once that happens, it will be virtually impossible to control how the embryos are used. That would make it impossible to enforce a ban on a baby cloning. People who stand with cloning insist that unimplanted balls of cells is not a person.
Created using Powtoon.